Good afternoon, everyone. It's a pleasure to be here, and I would like to thank the organization for letting me speak about our recent new point-of-care device, which is called the VeraSTAT. And I hope I will convince this it is not no snake oil. So this is a point-of-care device which is meant to be measuring antigen concentrations, typically proteins, in a fully quantitative fashion. It's a next generation system which is really second to none in terms of characteristics. And you will see that on the data I will be presenting on sensitivity, accuracy, ease of use, and cost efficiency. So typically bringing this point-of-care device to a next generation. It's operating from whole blood from very small volumes. And there is a possibility of multiplexing those, even though we have no operation today. The test is not ready for quantitative use, but it's coming. So what is the VeraSTAT? And you can see this in reality at our booth 306. It's a portable point-of-care analyzer, which is weighing 3.5 kilos. It's a 5-inch color touchscreen interface. The dimension, as you can see, is very small. It is operatable under a number of electricity powers. It runs with cathodic electroluminescence cartridges. The corresponding cartridges comes with ready-for-use identifiers, which are near-field communication cards. The system is doted with Bluetooth connectivity for sample result transfer. It's typically speaking English today, but this can also be moving in the future. Principle of testing, I said it's based on cathodic electroluminescence technology, which is, you've been using, most laboratorians have been using electrochemiluminescence, which is anodic in nature, from a very large manufacturer, but the technology is exactly the same. The cassette, it comes in cassettes, and you will see pictures of those, which are specifically labeled for the corresponding test, and which are using antibodies. And I will again show you some examples. It is currently pushed through the sample complex. The current is pushed through the sample complex, and you get an enhancement of the chemiluminescence thanks to this current. And basically, the chemiluminescence signal is measured, and an onboard microprocessor calculates the results of the analyte in the clinical specimen using a pre-programmed calibration point. And the result is displayed on the screen, but can be transferred as well to a Bluetooth system. How does it work in practice? You have, that's an example on an immunoassay, so sandwich typical immunoassay, which is built in. The excess of the labeled antibodies washed out by a built-in system in the cartridge. I will show you the cartridge afterwards. The electrical excitation comes afterwards and is read through a PMT system very classically. Whoops. Yeah. So that's a picture of the cassette. What you can see here is the entire cassette in a. It looks very big, it's very tiny. It's probably the size of my thumb here in total. What you have is on top, you have an NFC tag, which is indicating the lot-specific information of the cartridge. You have an electric system, which comes as a piston for the fluid that can be washed out. You have the sample application hole, and then you have two systems of membrane. You have a silicone membrane, which is typically holding the capture device, whether an antibody or an antigen. And then you have a second membrane, which is typically holding a dried up antibody system, which is itself labeled with the fluorophore or luminophore system. So as the system goes on, you put it on the machine, and then your sample is added in this small window that you have this square, the black square here. This application hole is typically holding a few microliters of sample. For example, our creatinine or CRP assay is measuring from 5 microliters of whole blood, and you get the signal measured after the extra signal has been washed away by this measuring buffer, and the amplification has been taken care of. The testing process for the user is very simple. Typically, you collect your sample. It's typically from a finger prick. It can be just picking up your finger, or you can take this from an anticoagulated blood system. You enter the patient ID, you scan your barcode, your NFC tag from the machine. Then you insert the cassette in the machine, and it is at that time that you input the corresponding sample. And you close the lid, you push the start button, and you get your results in a few minutes. Again, for this CRP assay I was alluding to, from this finger prick 5 microliters of whole blood, you will get your results in 6 minutes. And you get your result on screen, but you can also get your results sent out through Bluetooth to any kind of connection, so you can store it on a server. The machine itself will store up to 1,000 types of results. And of course, you can transfer them on a regular basis. Benefits, they are very obvious. You get your results very accurately in a very short time frame. 6 minutes is the average for the existing test menu. So it eliminates the need to send samples to a lab and facilitates immediate decisions. It's very simple and easy to use, as you have seen from the operation scheme. It's a very small footprint, lightweight, portable, can be used in a variety of locations and delivering diagnostic results when required. It's an all-in-one cassette. And as I said in the beginning today, what we have is uniquely having single test samples, but we are working on multiplexed applications, and there will be one very shortly on ApoE4 detection for the detection of Alzheimer's risks, for example. The test menu is being developed. The machine is being developed by a Finnish company that we have basically contracted with. And we are Randox is in charge of developing the test menu. So you will see in a few minutes what the test menu looks like. The results can be exported, as I said, by Bluetooth connectivity. The sample volume is very small, typically a few microliters. And it's coming with a built-in QC module, and the QC materials can be the one you would typically use in the lab for the corresponding analytes. The intention is to provide the same QC materials as the reagents become available. So typically, if you see these big charts, and you look at the last column, most of the low-cost items would come with lack of sensitivity. Obviously, you would know that the electroluminescence and that come with the biggest instrumentation are extremely robust in terms of accuracy and sensitivity, but would not come with portability or low cost. So we believe that this system does show a very nice combination of characteristics with high sensitivity as well as all the features you expect from a point-of-care device. So what is the test pipeline for the moment? We have some applications in veterinary biomarkers. The one we have today is for equine SSA, but all the other ones you have coming with a SAR are in development today. So foal IgG, feline SSA, camel SSA, canine CRP, cortisol, progesterone, and Rhodococcus equi. On the human IVD biomarkers today, we have 3 coming up, already available, which are the myxovirus-resistant protein A, as well as the CRP. Both are mostly used to discriminate between infection and a bacterial infection. And we have COVID-19 serological tests. I have one more slide on this one. Everything else is coming as in the next coming year and a half. A few information on potential performances that you can get from this device on CRP. So this one is not a high sensitivity CRP. It's meant for, again, discrimination of infectious disease. You get very nice precision of less than 11% for intra-precision, less than 13% for inter-assay precision CVs. That has been determined from liquid check, Bio-Rad QC materials. The limit of detection for this one is set to 5 milligram per liter. The assay range is 5 to 200 milligram per liter, and the traceability has been assessed using the WHO standards, which is quoted here. Interferences for most substances, that can potentially interfere have been tested, and these are the concentrations, the highest concentration that can be tolerated without any impact on those. You can see that the method comparison with a clinical laboratory method is very good and has been tested on a sufficient number of samples to be reliable. We have also a combination of serology profiles for SARS-CoV-2 antibody testing, using the nucleocapsid protein IgG testing. We also have the RBD system, which can be used for discrimination of SARS-CoV-2 virus infection versus vaccination. And we have one which is specifically looking at SARS-CoV-2 neutralizing antibodies. This is based on the original Wuhan strain, but we have, for research purposes, eventually, all of the corresponding proteins for all variants of concerns down to the Omicron BA.5. I think this is me for today. So if you have any questions, I will be happy to answer.